CK to YouTube.
All right, guys. So ventricles, pathway of cerebral spinal fluid. Okay. So I always got confused with this in school for some reason. I'm not sure why it didn't make much sense to me until later on when I was learning it. But all ventricles are is they're just little openings, excuse me, and pockets inside of the brain that allow for CSF to basically be stored, and also where it's created. Okay. So my biggest analogy for this is, you know, in the skull in particular, we have these things called sinuses. And essentially, what sinuses are is they're just little kind of openings or pockets where mucus is essentially formed or can be stored. Okay, maybe also when you have a sinus infection, what happens there is you get mucus that kind of gets stuck in there that has the bacteria or the virus that whatever we're carrying, whenever we have a cold or flu, whatever it is, and the bacteria just causes an infection in there, right? So maybe you feel your sinuses and maybe your cheekbones or maybe in your forehead. It just feels like a lot of pressure in there. It's a similar concept for the ventricles, okay? Except the ventricles are inside of the brain. So they're basically just the sinuses, the sini of the brain, okay? There are 4 of them total. We have 2 lateral ventricles, so they're basically just the first and second ventricle. We have a third, and then we have a fourth, respectively, okay? We're going to talk about these landmarks as they exist, right? So 2 lateral ventricles, we can see them here, okay? 2 of those guys right there, 1, 2. We have this landmark called the interventricular foramen, okay? So it's a hole that is between these ventricles, okay? As it's circled here. And what it allows for allows for the fluid, the CSF, okay, that is in the lateral ventricles to flow into that third ventricle, which we can see right here, okay? So it allows that to happen. To get from the third ventricle to the fourth ventricle, we're going to use this thing called the cerebral aqueduct, okay? And in the older days, 400, 500 years ago, what aqueducts were responsible for were transporting water, right? When we didn't have access to water pipes back then, we used aqueducts to help transport the water, okay? So similar concept, the aqueduct in the cerebrum is responsible for transporting the CSF to this fourth ventricle down here, okay? In the fourth ventricle in particular, there are a couple things. So the subarachnoid space, so this is where we are going to start to see the CSF start to infiltrate that space. And this is where we have all the CSF that nourishes, it bathes the brain, it keeps it hydrated. It's a really important space. Remember, and it sits between the arachnoid and the pia mater. Hence why it's called the subarachnoid space, because it's just below it, okay? And then we also have the central canal, which we'll talk about more next week, but it is that little opening that is in the dead center of the spinal cord, okay? And this is how CSF is able to travel through the spinal cord and nourish it, right? So to talk about the pathway, a couple steps here, okay? Step 1, CSF is secreted by the choroid plexus in each lateral ventricle. The choroid plexus is essentially this kind of red squiggly lines that we're going to be seeing throughout the entirety of this model. So that's the choroid plexus, all right? So CSF is secreted up here, step 1. Step 2, it flows through the interventricular foramina into the third ventricle, okay? So step 2, so it flows through that hole there, gets into the third ventricle here. Step 3, so the choroid plexus secretes more CSF, so it adds more to the mix, all right? So we can see that adding more, step 3. Step 4, CSF flows down the cerebral aqueduct, respectively. So it's going to follow this line down here as it travels towards that fourth ventricle, which is right here. Okay. When we get to step 5, what's going to happen? The choroid plexus here is going to add more CSF. So we're just continuously adding this stuff, all right? And then step 6, we can see these little holes here. And what these are called are apertures, okay? We have 2 lateral and 1 median aperture. And what they do is they allow for the CSF to exit so it can infiltrate that subarachnoid space, okay? So that's step 7. So the CSF exits through those apertures. Step 7 down here, we can see that CSF following the lines here. It enters that subarachnoid space, all right? So it can bathe the external surfaces of the brain and the spinal cord. And then lastly, we have the arachnoid granulations, which are these holes up here. Let's get a different color so you can see that a little bit better. We have these holes up here at the top of the brain in the subarachnoid space that allows for that CSF to be reabsorbed into the venous blood flow. The venous blood flow is this darker blue structure that has these blood vessels extending into the brain. CSF goes in there so it can get recycled, and then the whole cycle happens over again. So we start back at step 1. Okay. So really straightforward, okay? The ventricles are just little openings, pockets in the brain that allow CSF to be created so it can sit, it can flow, bathe the brain, hydrate it, nourish it, all that stuff, all right? That's all it is. And it follows these 8 steps in order every time, okay? Okay, so with that, moving on to the sheep brain, okay? Remember, big tip once again, the pictures are helpful, but remember, the pictures will not show up on our celebration. It is going to be the live sheep brains themselves, okay? So everything that we want you to be able to identify is in number 4 right here, okay? So all of these structures, and none of this is new stuff. We talked about this in the models up above, the figures up above. This is all stuff that you guys were able to identify on the sheep brains. I walked around to the groups and helped point some things out to you. But if you need more time with this stuff, remember, this is always down on the lab for you to take a peek at, so you can find it, so you can demolish when you get to these questions on our celebration, okay? So, looking here, okay, dorsal view. So looking from the top, looking basically from an eagle's eye view, okay, dorsal view. We see the cerebrum, upper part of the brain, longitudinal fissure right down the center, left and right halves, okay. Don't need a virus or threat protection warning there. Transverse fissure, all right, separates, remember, cerebellum, this guy right here, cerebellum, separates it from the cerebrum up here. So that line, what it's going to look like on my sheep brain here is it's going to look like this. Okay, kind of has like a hill-looking structure to it, right? That's that right there. Olfactory bulbs, so we have to flip it over to the ventral surface. Remember, olfactory bulbs, okay, there's 2 of them. They kind of have a flattened looking appearance, as you can see. They always sit up towards the top, so away from the brainstem. Optic chiasm sits right in the center, and maybe you could see a little bit of that crossing that undergoes, okay? Corpus callosum, we'll see that better on the lateral view. Lateral ventricle, lateral view, thalamus, yep, so all this right here is lateral view. Here we can also see the midbrain, okay? So midbrain, remember this is the first of the 3 spinal, the brainstem structures. Always start from the midbrain, always situates itself at the top. Then the pons, you can see a little bit of that extra lumpy looking structure, that's always the pons. Everything below that is the medulla, okay? So everything below that's medulla. So midbrain, as you remember, has a couple extra structures. If we scroll down a little bit, what's happening here is we're looking from the posterior view, so from a caudal view. What this person's doing here is they're pulling down, they're retracting that cerebellum so we can better see that superior and the inferior colliculus of the midbrain here, okay? Deep inside, okay, deep inside of a little pocket here is that little tiny pineal gland, which is better visualized also on the lateral view, okay? But this is the best view for identifying those superior and the inferior colliculi, okay, of the midbrain. So good there. Scrolling down here. Okay, lateral view, a sagittal section, so we cut straight through that longitudinal fissure, and this is what half the brain looks like, okay? Corpus callosum, we can see that white matter right here, okay? That white matter, all right? White matter there connects the left and the right side of the brain, okay? So they can communicate with each other, okay? Lateral ventricle, so it's an opening here, so let me actually erase my eraser. Eraser, perfect. It's actually this little slit, that opening that we can see here, where the CSF is starting to be housed and created, okay? The slit opening there, okay? Optic chiasm right here, okay? White little structure right down there. Thalamus is always right in the center, as we talked about, and hypothalamus is right below it. So thalamus above, hypothalamus right below. The cerebrum is just the entirety of the upper brain. Pineal gland always situates itself right in the back. You can see it a little bit on that retracted view up here, but I always think you see it better on the lateral view when we're looking at a sagittal cut. I always think it reveals itself a lot better. Erase it there. Superior and inferior colliculus, some of them on the sagittal view, it's very hard to see, which is why I once again prefer this exposed view. When we retract that cerebellum, they look so much more better there. Way better for you to identify. 3 structures of the brainstem, always midbrain here, connects with the colliculi. The pons, you can see a little bit of that lump right there, and then always right below that's the medulla, okay? 4th ventricle, remember a little slit inside of that cerebellum there, very, very small. cerebellum, as it's identified, right below the cerebrum, and then the arbor vitae. Remember, okay, the tree of life. All right, you see that white matter? It looks like a little tree extending out with the branches. That's the arbor vitae, the tree of life. Okay. Okay, guys, besides that, it's just labeling. That's all you guys got left. The review section, of course. So, like I said, this is a decently quicker lab. There's a lot of models and stuff we want you to know, and I think you guys are going to be just fine with this stuff, okay? So remember, this lab, lab 10, and then lab 11 are combined for our next celebration, which is in 3 weeks. So it's after Thanksgiving break. So we have a lot of time, okay? So make sure we come back and we review this stuff, all right? Just a little bit. Bite at that whale, as I always say, okay? So I definitely think we did all of these things, okay? I think you guys are going to be able to answer these questions if you were shown them. You have these videos at your disposal. You have the lab manuals, and if you have any questions on anything, feel free to reach out. Besides that, TK is out. All right, guys. See you next week.